Alright, welcome back, fellow scientists. So if you'll remember last video, we talked about the process of transcription. And remember, that was the first step in taking the information in DNA and putting that to work, using that to make the workers, using that to make proteins, which then control the cells. Right? So the second step that we're going to talk about today is translation, the second step of putting the information in DNA to work. So let's go ahead and write down a definition of translation. So translation is taking the information in RNA and using that to make proteins. Taking the information in RNA and making proteins. Which, remember, proteins are the workers in our body, so they actually do stuff. They control what we look like, what goes on, cellular respiration, photosynthesis in plants, all sorts of stuff like that. So we could summarize translation. It's simply taking the information in RNA and using that information to make proteins. Alright, now before we get into the process of translation, we talked about the three different types of RNA. Last time, I want to go a little bit more in depth so that we can understand the pictures that I'm going to draw during the process. So we'll start with mRNA, which is the most famous type of RNA, and it's single-stranded, as we talked about before. DNA is usually double-stranded, mRNA is usually single-stranded, and then we'll write some nucleotides of RNA. So we'll write A, U, G. Remember, in RNA we don't have a T, we have a U, and then C, C, G, and these are just random. A, U, U, they could be anything. G, C, U, G, C, U, and then UGA, UGA. Perfect. And all of those are U's. U, U. Okay, good. Sweet. So that's what mRNA looks like. Now, mRNA is broken down into three-letter groups called codons. Right, so there's a codon, there's a codon, there's a codon, and we'll see why that's important later on. So, quiz question, how many codons are in this piece of mRNA? If you said 5, you're right, we already did 3, and then we can make 4, 5 codons. Alright, so that's mRNA. Now, rRNA stands for ribosomal RNA. And the ribosome, actually, we just drew them as dots when we were looking at the cell, but a ribosome actually has two parts. There's a small part, and then there's a large part. So that's a ribosome. And the rRNA actually kind of lives in the ribosome. There's 3 pieces up here, and then there's 1 piece down here. And the rRNA, it just helps match up the tRNA and the mRNA. M-A-T. Match up the tRNA and rRNA. So that's the rRNA. And then the tRNA. This is kind of funky. This looks a little bit weird. The way that it's usually drawn is something like this. We go down here, and there's kind of like a doorknob looking like thing that's off the page, and then we go out here like that, and then up. And there are base pairs that are holding this together. So A pairs with U, C pairs with G, holding that together, holding that together. These in these loops, these are unpaired bases, so these are not complementary base pairs. And then up here on our tRNA we have an amino acid. Remember, amino acids are the building blocks of proteins, so if we have a bunch of amino acids all in a row, then that's going to make a protein. So that's an amino acid right there, and again we have base pairs here, unpaired bases here, base pairs right here holding these together, and then here we have our most important bases of our tRNA. These are 3 bases, and these 3 bases are called the anticodon. Actually, you know what? I'll make this a little bit more specific. So let's say that this is UAC. Alright, so these would be our bases right here, and these bases are called our anticodon. And what this does is eventually, during the process of translation, our tRNA, our bases on our anticodon, UAC, are going to match up with our mRNA, our bases on our codon. And as you can see, I made it so that UAC is complementary to AUG, UAC, right? So this anticodon right here is going to, match up, is going to complementary base pair with this codon right here, and it's going to be facilitated, it's going to be helped by the ribosome. Alright, so let's look at the process of translation then. So we'll have, and this is going to be kind of quick, but there are other videos that do it a lot better because it's a process, and I'll direct you to those. But we're going to have our mRNA, so we're going to have AUG, what was it, CCG, something like that. And so this is going to be before translation. TRANSLATION. Before translation. That's horrible. TRANSLATION. Before translation. Alright, so before translation, we're just going to have our mRNA, right? Then during translation, we're going to have our mRNA, and we're going to, for the sake of trying to be accurate, we're going to kind of zoom out. So we're going to have our mRNA, and we're going to have our ribosome, the small part and the big part, and then we're going to have our tRNA. That's going to come, and it's going to be like that. And then we're going to have a little amino acid right there, and then this is going to be kind of like that, right? And so we're going to have a little amino acid right there, and its anticodon is going to match up with this codon, and then we're going to have another tRNA kind of like that, and this is horrible, by the way. And then we're going to have another kind of little arm like that, and we're going to have another thing like that, right? And so there's actually 3 tRNAs in the ribosome, but I didn't do a good job of drawing it. But the basic idea is that these anticodons match up with these codons, and there's a specific amino acid attached to each tRNA. And so there's a specific amino acid for each codon that matches up to each anticodon, right? And then depending on the order of the codons, depending on the order of the bases, which is determined by DNA, then this amino acid is going to be transferred to that amino acid, so that after that we will have, again, mRNA, and then our ribosome is going to be moving down the strand, kind of like this. So then we'll go like that. We'll go, actually that's horrible. Delete. We'll go like that. And we'll go like that. And so then this tRNA right here, this is now outside the ribosome, and it's empty. Right? It doesn't have an amino acid right here. This tRNA right here, it now has 2 amino acids because this amino acid was transferred onto here. So then we're going to have another tRNA. Oh, and by the way, there's of course anticodons. So then we're going to have another tRNA come in, and it's like that, and it's going to have an amino acid, and then we're going to transfer these 2 onto that one, and so on and so forth. And then the ribosome is going to move down, and then this tRNA is empty and it moves out, so that eventually, after the process of translation, we're still going to have our piece of mRNA. Our ribosome is no longer going to be attached. It's going to be separated into two different pieces that can come together on the same piece of mRNA or a different piece of mRNA. We're going to have a whole bunch of empty tRNAs right? Notice no amino acid right here. So we're going to have a whole bunch of these. And I'm going to go ahead and cheat. So we're going to have a whole bunch of empty tRNAs. Kind of like that, right? And then we're going to have our end, our goal, we're going to have a specific chain, a specific string of amino acids, right? If we have a specific string, a specific chain of amino acids, we know that that is a protein. And that's the whole entire goal of transcription and translation, because as we know, the proteins are the workers. They do stuff. They control the cells. So that's the process of translation.